Neurolemmocytes. Schwann cells, which were named after physiologist Theodor Schwann, or neurolemmocytes, are the principal glia of the peripheral nervous system. Glial cells function to support neurons, and in the peripheral nervous system also include satellite cells, olfactory and sheathing cells, enteric glia, and glia that reside at sensory nerve endings, such as the Panitian corpuscle.
I want to talk about Schwann cells. Schwann cells are glia of the peripheral nervous system, derived from neural crest cells, and named after a person who described them. Schwann cells come in a couple of shapes. Some are fairly shapeless cells that have little troughs on their surface, and the axons of neurons that have small diameter axons often just sit inside these troughs. So these are neurons with the soma and leaving off the dendrites, and these have small axons, small diameter axons, and they'll just kind of sit in these little troughs on the surface of these Schwann cells, and these are called non-myelinating Schwann cells. They do provide some support to these peripheral neuron axons that are small diameter, but they don't myelinate them, so they're non-myelinating Schwann cells. Now, peripheral neurons that have a larger axon, let me just draw this one with a little larger axon, usually have a myelin sheath. And just like in the central nervous system, there'll be a sheath going all the way down the axon that's regularly interrupted by these little gaps called the nodes of Grandier. And these little segments of myelin in the myelin sheath of peripheral neurons has the same structure and function as the myelin sheath in central neurons. However, the Schwann cells create the myelin sheath in the peripheral nervous system, as opposed to the oligodendrocytes of the central nervous system. Now, while the structure and the function of the little myelin segments is the same, a big difference between the Schwann cells in the peripheral nervous system and the oligodendrocytes in the central nervous system is that a Schwann cell only produces the myelin sheath for one segment of only one axon. It's not myelinating multiple neurons like oligodendrocytes do. Now, let's take a little closer look at one of these areas of myelin sheath, and let's cut through this segment of myelin just like this, and we'll look at it this way, like we're looking down from the end of the axon. And this is going to look just like the myelin sheath for a central axon. Here's the axon, and we're looking at it end on, and the cell membrane of the Schwann cell, which is this material we call myelin that's very rich in lipid, is just going to be wrapped again and again and again, very thinly and very tightly, like a roll of tape around the axon. Now, for a Schwann cell, this is almost the entirety of its cell membrane.
Basics. There are two types of Schwann cell, myelinating and non-myelinating. Myelinating Schwann cells wrapped around axons of motor and sensory neurons to form the myelin sheath. Schwann cells are involved in many important aspects of peripheral nerve biology: the conduction of nervous impulses along axons, nerve development and regeneration, traffic support for neurons, production of the nerve extracellular matrix, modulation of neuromuscular synaptic activity, and the presentation of antigens to T lymphocytes.
What you will see and hear in this presentation is the development of myelin in the peripheral nervous system and the propagation of the action potential along a myelinated axon. Schwann cell forms a protective covering around the axon. Schwann cells start to develop in the embryo and continue to increase the wrapping around the axon through childhood. This development increases the thickness of the wrappings, which peaks in adolescence. This is why teenagers have such quick responses.
What are they? Schwann cells are a variety of glial cells that keep peripheral nerve fibers alive. In myelinating axons, Schwann cells form the myelin sheath. The sheath is not continuous. Individual myelinating Schwann cells cover about 100 micrometers of an axon, which can be up to a meter or more in length. The gaps between adjacent Schwann cells are called nodes of Grandier. The action potential jumps from node to node in a process called saltatory conduction, which can increase conduction velocity up to 10 times without an increase in axonal diameter.
The axon is not covered by the neuroglial cell. These spaces are called nodes of Rambier. From what you already know, action potentials occur at the axon hillock and continue to be repeated away from the cell body, much like dominoes falling one after another. An action potential starts on a polarized membrane, which is -70. A stimulus causes the sodium gates to open slightly, and sodium starts to trickle into the cell. If the cell reaches -60 or threshold, the sodium gates open wide and sodium floods in, bringing the inside of the axon to +30. At this point, the sodium gates close and potassium gates open. Potassium starts to pour out of the cell. This allows the neuron to become polarized again. Then the sodium-potassium pump starts to actively transport sodium out and potassium back into the neuron. First, we will look at the propagation of the action potential in the unmyelinated axon. Propagation is the repeating of action potentials down the axon. The action potential is repeated because as the sodium comes in, it diffuses to adjacent areas within the axon. As the sodium increases in this area, threshold is reached. Sodium gates open wide, sodium rushes in, causing depolarization and an action potential.
Functions. Non-myelinating Schwann cells are involved in maintenance of axons and are crucial for neuronal survival. Myelinating Schwann cells begin to form the myelin sheath in mammals during fetal development and work by spiraling around the axon. A well-developed Schwann cell is shaped like a rolled-up sheet of paper, with layers of myelin in between each coil. The inner layers of the wrapping, which are predominant. Functions. Non-myelinating Schwann cells are involved in maintenance of axons and are crucial for neuronal survival. Myelinating Schwann cells begin to form the myelin sheath in mammals during fetal development and work by spiraling around the axon. A well-developed Schwann cell is shaped like a rolled-up sheet of paper, with layers of myelin in between each coil. The inner layers of the wrapping, which are predominantly membrane material, form the myelin sheath, while the outermost layer of nucleated cytoplasm forms the neurolemma. Only a small volume of residual cytoplasm communicates the inner from the outer layers.
Compare the unmyelinated axon with the myelinated axon. You can see that action potential reached the end of the myelinated axon more rapidly than the unmyelinated axon. The speed of the propagation is faster going from node to node than action potentials that develop adjacent to the previous action potential.
Lineage. Schwann cells are of neural crest origin. During most embryonic development, neural crest cells first differentiate into Schwann cell precursors at around embryonic day 12 to 13. These precursor cells subsequently differentiate into immature Schwann cells at approximately embryonic day 15 to 16, persisting until birth. The postnatal fate of the immature Schwann cell depends on its random association with axons. Immunoreactivity. Different classes of Schwann cell express characteristic antigenic markers that can be targeted with antibodies. Myelinating Schwann cells can be visualized by immunohistochemistry using antibodies against the proteins S100, myelin protein 0, and myelin basic protein. Non-myelinating Schwann cells, such as those that form Brimac bundles and terminal Schwann cells, are positive for S100 and glial fibrillary acidic protein. Neurolemmocytes transplantation and regeneration. A number of experimental studies since 2001 have implanted Schwann cells in an attempt to induce remyelination in multiple sclerosis afflicted patients. In the past 2 decades, many studies have demonstrated positive results and potential for Schwann cell transplantation as a therapy for spinal cord injury. both in aiding regrowth and myelination of damaged central nervous system axons. Indeed, Schwann cells are known for their roles in supporting nerve regeneration. Demyelinating diseases of the nervous system. This group of diseases is characterized by lesions that are associated with loss of myelin with relative sparing of axons. There are many types of diseases that damage myelin in concert with the destruction of axons. However, these conditions are not considered demyelinating due to their non-specific and secondary effects on myelin. Additionally, there are metabolic disorders of myelination characterized by the accumulation of abnormal breakdown products. These are considered to be dysmyelinative disorders. Guillain-Barré syndrome. This is a dramatic acute demyelinating neuropathy with rapid onset, which is hours to days. This usually produces weakness on the extremities and axial musculature, which can evolve to respiratory motor failure and asphyxiation if support is not available. Prior to availability of artificial respiratory support, the mortality rate was 60%. Involvement of the autonomic nervous system also may occur, indicating an axonal involvement, and indeed there is a form of this syndrome in which axonal involvement predominates. Summary. The Schwann cells are a type of cell throughout the entire peripheral nervous system. This system includes all nerves going out to muscles as well as sensory nerves coming from the muscles back to the spinal cord. Schwann cells are a type of support cell in this system. Some important points about Schwann cells are: Schwann cells insulate individual nerve fibers, which is necessary for sending appropriate electrical signals throughout the nervous system. Schwann cells are not stem cells. They are adult cells that can only be Schwann cells. Schwann cells are absolutely essential for regeneration in the injured peripheral nervous system.